So, dear students, today we will speak about the meninges, about the circulation of cerebrospinal fluid and blood supply of the brain. This is quite a big topic, so I will focus on clinical aspects and the details you will learn in the practices. So let's start with the meninges. Here you can see a schematic drawing which shows the three layers of these membranes which cover the brain. Actually, it is similar to the meninges of the spinal cord. So the outermost layer is called dura mater. This is a very thick layer composed of dense connective tissue, and in contrast to the dura mater of the spinal cord, it is a fused layer of the periosteum and the dura mater itself. So we do not have a gap between these two layers. And it is now shown in yellow, yes, and you can see that at some places it forms folds, for example, here on the left side, and you will also see that the dura will form the wall of the venous sinuses. The inner layers are the arachnoid and the pia mater. The pia mater is directly on the surface of the brain. It follows the surface of the brain. It invaginates into these grooves as well. And the arachnoid, it follows only the surface directly under the dura mater. And we will also speak about these arachnoid granulations, which will be important for the reabsorption of the cerebrospinal fluid. We have two virtual spaces between the dura and the bone, the cranial bone. This is called epidural space. Actually, it doesn't exist only if we have a mass, a hemorrhage, for example, in this space. Or between the arachnoid and the dura, we speak about subdural space. It is only visible if we have a bleeding there. But the space between the arachnoid and the pia mater is a very important space that is called subarachnoidal space, and here we can find the cerebrospinal fluid, which is very important. It makes the brain more lighter, so the brain is actually swimming in this fluid. And as you can see, we also have vessels in the subarachnoidal space. And the pia mater and the arachnoid are connected with each other by so-called arachnoid trabeculae. These are composed of collagenous fibers, but as you can see in this schematic drawing, on the surface, we have specialized fibrocytes called meningeal cells, which actually line the subarachnoidal space. And also on the innermost surface of the dura, this is now shown by this red line, we also have closely packed modified fibrocytes called also meningeal cells, and it forms the neurothelium. In the next picture, we will also see it. Yes, this picture shows the histological appearance. You can see that here on the surface of the brain, we have a glial limiting membrane and also a basal membrane, and then we have the meninges. The outermost layer is the dura. So as I mentioned, this is a It contains of two layers, an endosphere layer and a meningeal layer. And actually, this is a very dense connective tissue. And on the innermost, so the inner layer is covered by this modified fibrocytes called meningeal cells. And this represents actually a blood liquor barrier, so blood CSF barrier between the meningeal vessels and the subarachnoidal space. And then in green, you can see the arachnoid and the pia mater and the connections, these arachnoid trabeculae as well. And both layers are composed of loose connective tissue made by collagen fibers. And here you can also see this meningeal cells, so these modified fibrocytes which then line the subarachnoidal space and also surround the subarachnoidal vessels. This subarachnoidal space continues actually in form of small perivascular spaces inside of the brain. So the vessels which enter the brain are followed by this, by the pia mater and by these small fine canals called Virchow-Robin spaces. And these are also clinically important because they can be enlarged, and these dilated Virchow-Robin spaces must be distinguished from neurodegenerative diseases. So in multiple sclerosis, for example, we have lots of these dilated spaces. Normally, some dilated spaces can also occur. The dura and also the other meninges are supplied mainly by the branches of the middle meningeal artery. You can see here in these pictures how the dura looks like. So this is like a cap around the brain, and these meningeal vessels are located on the outer surface of the dura. So they actually run in the epidural space, and so the most important artery is the middle meningeal artery coming from the external carotid, but we also have an anterior and a posterior meningeal artery. But as you can see, only the anterior one comes from the internal carotid. So the meninges are mainly supplied by the external carotid artery. And if you remember the skull, we can see this sulci arteriosi on the inner surface of the calvaria, which actually are formed by the branches of the meningeal arteries. And clinically, we can here mention the epidural hematoma. It means if we have blood, if we have a hemorrhage between the dura and the skull, as you can see in this picture, and it is a type of traumatic brain injuries followed by fracture of the temporal bone. Mainly, and this is a life-threatening situation because the blood extends then, and if you make a CT picture, you will see a lens-like biconvex appearance of this blood, because the expansion stops at sutures where the dura is fixed to the calvaria, and then the bleeding expands then toward the brain tissue, and so it causes an increasement of the intracranial pressure. The dura is innervated mainly by the trigeminal nerve. As you can see here in this picture, all the three branches of the trigeminal nerve has meningeal branches, but also the vagus nerve, here as you can see in the posterior cranial fossa, and also the upper cervical nerves give meningeal branches. And it is interesting that these meningeal branches are also found in the wall of the meningeal vessels. And if you have a headache, it is actually the tension of these meningeal vessels. So these meningeal vessels have also a sensory innervation. And the vessels have naturally also a sympathetic innervation for the contraction, and it comes from the spinal cord and via the superior cervical ganglion. The postganglionic fibers surround the carotid arteries, and so they can reach the vessels. But the brain tissue itself has no pain receptors, so you can actually make operations when the patients are not anesthetized. Clinically, we can mention the neck stiffness. This is the leading symptom of the meningitis. It belongs to the meningisms together with the photophobia and headache. And this is actually reflex, a visceromotor reflex, which causes the refractory contraction of the nuchal muscles, and then the neck cannot be bent forward. And you can see the reflex arc, so the sensory branches coming from the meninges can activate motor neurons, the axons of which innervate the neck muscles, and you can see this typical sign. And in the diagnosis of the meningitis, this is important, and this is because the meninges are also innervated by the cervical spinal nerves. Yes, afferents coming from the cervical spinal nerves and efferents go to the neck muscles. So the dura forms so-called folds, and you can see schematically and also in a preparation the most important folds. Falx cerebri separates the two hemispheres from each other and goes into this longitudinal cerebral fissure. This is the largest dura fold, and perpendicular to this, we have the tentorium cerebellum, which separates the cerebellum from the occipital lobe and fills the transverse fissure. It has a crescent shape so that the brainstem can go through here in this part. The falx has a sickle shape typically, and then we have the continuation of the falx under the inner occipital protuberance. It is called cerebellar falx, which separates the two hemispheres of the cerebellum from each other, but this is a very small fold. And we have two more, the trigeminal cave and the sellar diaphragm. These are folds you will see later in the practices. And as you can see, the venous spaces are also formed by the dura. I will show it right now in the next picture. But one more clinical aspect regarding these folds, if you have increased intracranial pressure due to a tumor, hemorrhage, or hydrocephalus, or a mass in the intracranial space, it can lead to herniation. It means that the brain tissue will be shifted into a foramen, for example, into the foramen magnum. The cerebellar tonsils can be shifted into this, or under the tentorium, the temporal or the occipital lobe. You can see that these are now shifted beneath the tentorium, and this is a life-threatening situation, and it causes the compression of vital centers of the brainstem. In this picture, you can see the largest venous sinuses, dural sinuses. These are large venous spaces actually formed by the dura mater, which drain the venous blood into the jugular vein. As you can see in this schematic drawing, these dural sinuses are also lined by endothelium, but otherwise the wall is formed by dense connective tissue like the wall of the dura, and this is inelastic and can that's why resist the intracranial pressure caused by the brain and the liquors or the cerebrospinal fluid and the vessels. And you can see that in the falx, you have in the upper margin of the falx the superior sagittal sinus, in the lower margin the inferior sagittal sinus, and where the falx and the tentorium meets, then you have the straight sinus. This is a sagittally arranged sinus. And then you have also heard about the transverse and sigmoid sinuses in the first semester, and when you discuss the skull, because these sinuses have a direct contact with the bone and form grooves on the inner surface of the calvaria and the skull. And at the end, the venous blood is drained then to the internal jugular vein. And in this picture, you can also see the smaller sinuses. Here, for example, the sphenoparietal sinus, or we also have sinuses on the petrosal bone, superior, inferior petrosal sinuses. And I want to emphasize here in the middle part of the base of the skull, the cavernous sinus. You will learn about it in details in the practices, but as you can see here, it surrounds the pituitary gland and is located on both sides of the body of the sphenoid bone. And the carotid artery runs through, and also lots of cranial nerves. So clinically, it's a very important space. It has a connection to the ophthalmic veins and pterygoid plexus as well. And clinically, it is very important. For example, it can come to cavernous sinus thrombosis, for example. That means formation of blood clot within the cavernous sinus as a consequence of an acne or sinusitis or dental infections or middle ear infections, because due to this bacterial infection, so this bacteria can spread toward the cavernous sinus because we have an anastomosis between the facial vein and the ophthalmic veins. And as I mentioned, the ophthalmic vein has a connection to this sinus. So this is a dangerous condition, and this is that dangerous triangle where the infections can cause this condition. Or another case, the sinus cavernosus fistula, it means that the internal carotid artery ruptures inside of this cavernous sinus, and that's why the arterial blood will actually enter the sinus, and as a consequence, the venous return will be impeded, and we will see a very interesting sign. The eye will pulsate, and it is called pulsating exophthalmos. But let's go further. Here in this picture, you can see again this. So some of these venous sinuses, the superior sagittal, and here the straight sinus. And this picture shows how the cerebrospinal fluid can reach these sinuses. So this fluid is produced by the choroid plexus. And you have already heard about the ventricles and the choroid plexus. And the arrows show how the cerebrospinal fluid flows, and here in the fourth ventricle it can leave the ventricles. We have altogether 3 openings: a median aperture called foramen of Magendie and 2 lateral apertures called foramen of Luschka. These are not visible now, and then the cerebrospinal fluid will enter the subarachnoidal space. And here, where the subarachnoidal space is enlarged, we speak about cisterns. Yes, so cisterns are enlarged subarachnoidal spaces. We have more cisterns. I have mentioned here some of them. Yes, the largest ones. You do not have to know all of these cisterns, but some larger cisterns you have to also know by names. And then if you follow these arrows, the cerebrospinal fluid will also descend behind the spinal cord, and then in between the denticulate ligaments, it can flow into anterior direction, and then it can ascend. And at the end, the fluid will reach these arachnoid granulations, where it will be reabsorbed into the venous blood of the venous sinuses. About the choroid plexus, I do not want to speak in details now. You have previously heard about it, but I show only a schematic drawing for this. As you can see, the ventricles are lined always by ependymal cells, and where the vessels of the pia mater protrude, these ependymal cells, we have then the choroid plexus. Otherwise, we speak about tela choroidea. And here in this area, the capillaries are fenestrated, but we have lots of tight junctions between the ependymal cells. So this is actually the inner blood liquor barrier. And the cerebrospinal fluid is actively produced by the ependymal cells, so they produce sodium, and the sodium drains then water. And the tinea, I want to mention the tinea are sites for fixation of the choroid plexus, and in the practice you will learn these sites. So now at the moment you won't understand this picture, but later you will learn where the choroid plexus is fixed in the lateral ventricle. Here you can see the central part, for example, of the lateral ventricle, or the inferior horn of the lateral ventricle. We also have choroid plexus, and in the third and fourth ventricles we also have choroid plexus. This picture shows these arachnoid granulations, called also arachnoid villi or pachyony's granulations. These are protrusions of the arachnoid, as you can see here, into the venous sinuses, mainly along the superior sagittal sinus. We have lots of arachnoid granulations, and as you can see, these are covered by sinus endothelium, and the reabsorption takes place through transcytosis. And this is the main way for the reabsorption, but also via capillaries of the pia mater and along the perineurium, the cerebrospinal fluid can be also absorbed. And clinically, I want to mention the meningioma. This is actually a benign tumor arising from the arachnoid villi. It can grow very intensively, so it can be huge, but it is a benign tumor, so it can be removed. And in the diagnosis, the angiography is a very important method, because with this method we can see that these tumors are mainly supplied by the external carotid artery, in contrast to a brain tumor, which are mainly supplied by the internal carotid artery. Papillar edema means the swelling of the optic disc, and this is also a sign of if the intracranial pressure is increased. So tumors, bleedings, other masses can cause the increasement of the intracranial pressure. And as you already have learned, the subarachnoidal space and the meninges follow the optic nerve as far as the eye bulb. So this increased pressure has also an effect on the papilla, and in this case the papilla has not a sharp border, but this you can see this swelling around this papilla. Hydrocephalus is the condition if the cerebrospinal fluid is accumulated in the ventricles, it cannot be absorbed, or we have a blockage somewhere. This is the so-called obstructive form. In this picture you can see a tumor of the pineal gland, pineal gland has a direct contact to the cerebral aqueduct. So in this case, the cerebrospinal fluid cannot enter the fourth ventricle, and that's why the ventricles will be enlarged. Or we can have another type if you do not have any obstruction, but the arachnoid granulations are functionally inactive, so the fluid cannot be reabsorbed, and it can happen after a meningitis or subarachnoidal hemorrhages. In adults, it will lead to the increasement of the intracranial pressure, but in children, when the head can be enlarged, then you will see this typical shape of the head, the progressive enlargement of the head in case of hydrocephalus. Okay, let's go further to the blood supply of the brain. This schematic drawing on the right side shows the most important arteries, but in details you will discuss these branches in the practices. I only want to emphasize that the brain is practically supplied by two systems, by the internal carotid artery and by the vertebral artery, but these two systems are connected with each other, and this is called Willis circle. And these branches you can see now run actually in the subarachnoidal space. That means where we also have the cerebrospinal fluid. So if you have a rupture of these arteries, which happens in case of an aneurysm, for example, if an aneurysm ruptures, then we will have a subarachnoidal hemorrhage. So that means that in case of a subarachnoidal bleeding, we have the rupture of these aneurysms. So in this case, you will find blood in the liquor if you make a lumbar puncture. And this is a very severe condition with a rapid onset of a very strong headache. And it requires surgical treatment naturally. These following pictures will show which areas of the brain are supplied by the different arteries, starting with the rhombencephalon, so cerebellum. You will learn it in details in the practices how the cerebellar arteries can supply the cerebellum, which are these branches. The pons is mainly supplied by the basilar artery, the medulla oblongata by the vertebralis, by the vertebral artery. And if you consider the cerebrum, yes, these pictures show the outer surfaces, so the convexity, and here the medial surfaces. And these colors, if you follow these colors, you will see that the middle cerebral artery supplies almost the entire surface of the convexity, but these medial margins of the frontal, parietal, and lobes are supplied by the anterior cerebral artery and also the medial surfaces together with the corpus callosum. And the posterior cerebral artery supplies the entire occipital lobe together with these marginal parts of the temporal lobe, and also the medial surface of the temporal lobe is supplied by the posterior cerebral artery. And in this horizontal and frontal sections, you can follow how the inner parts of the brain, so the white substance and the basal ganglia are supplied. And I want to emphasize here the middle cerebral artery, and its branches, the lenticulostriate and thalamostriate branches. These are shown here by this reddish color here. And you can see that the main parts of the basal ganglia, so striatum, caudate nucleus, and also partly a little bit the pallidum supplied, and main part of the capsula interna, so this internal capsule, are supplied by the middle cerebral artery. And clinically it will be very important because this is the most common site of stroke here, this part of the internal capsule and basal ganglia. And we have a branch of the middle cerebral artery, or it can directly come from the internal carotid, called anterior choroid artery. This is also an artery which corresponds, which gives branches to the internal capsule and the basal ganglia. The posterior cerebral artery is important in the blood supply of the diencephalon, so thalamus, hypothalamus, and also blood supply of the hippocampus, and it also gives branches to the choroid plexus, like also the anterior choroid artery. These are the two arteries which supply the choroid plexus. And also the midbrain will be supplied mainly by the posterior cerebral artery. Clinically, this stroke is a very important condition, and it can occur after an ischemic blockage caused by thrombus or an embolia, or by a bleeding, by hemorrhages, as you can see schematically in these pictures. And this is now a so-called intracerebral hemorrhage. Yes, we can have more types of intracranial hemorrhages. We have already mentioned this epidural hemorrhage caused by the rupture of the meningeal arteries, or we also have mentioned the subarachnoidal bleedings. And I will also speak about the subdural bleeding right now, but now you can see the intracerebral hemorrhages, and it occurs mainly here, where we have the internal capsule and the basal ganglia. So this is a predilection site for this intracerebral hemorrhage, caused by the rupture of the branches of the middle cerebral artery. As you can see, these branches originate perpendicularly from the middle cerebral artery, and that's why the vessels can be injured here. In the background of this bleeding, we have mainly a high blood pressure or arteriosclerosis, but we can also have non-hypertensive types, but this is a rare condition. I also want to emphasize that we have collaterals between the branches of the external and internal carotid arteries, or branches, or between the branches of internal carotid arteries of the two sides. You can see here some examples for this anastomosis. For example, here in the medial angle of the eye, the facial artery forms an anastomosis via angular artery with the ophthalmic artery, or the superficial temporal artery can also form an anastomosis here. Or between the occipital artery and the vertebral artery, we can have an anastomosis, or actually the Willis circle itself is also an anastomosis, but this anastomosis can only take over the blood supply of other brain areas if the occlusion develops slowly. So in acute occlusion, they cannot help anymore. Yes, in this picture shows some variations of the internal carotid artery. This is not important for the exam, but if you are interested, you can have a look at these pictures. But we will jump further. And at the end, I want to speak about the veins, about the venous drainage of the brain. And this picture shows the arachnoid layer here. And under the arachnoidea, we will see lots of veins, both on the convex and also on the inferior surface of the brain. And these are the subarachnoidal veins which collect the blood from the surface of the brain. And naturally, we also have veins inside of the brain. And we will see that these subarachnoidal veins are connected with the sinuses by so-called bridging veins, and the sinuses will lead to the internal jugular vein. This picture is a schematic drawing now about the most important sinuses, shown by this blue color and by the internal veins of the brain. So in green you can see the most important internal veins, internal cerebral vein, which fuse with the basilar veins, and they form the great cerebral vein, which will drain into the straight sinus. And in purple you can see the most important superficial veins, which will then enter the neighboring sinuses. And at the end, the blood will be drained into the internal jugular vein. Here on the right side, you can see again the cavernous sinus and its connections to the other important veins. Some words about the bridging veins. Yes, bridging veins are shown here in this schematic drawing, and as you can see, these are actually the connections between the superficial veins and the venous sinuses. So they will pass the dura and to be able to enter into the sinuses. And if we take the dura and if we expand it, we will see these bridging veins also in our preparations. And do not mix the bridging veins with the emissary veins. Yes, the emissary veins are connections between the sinuses and the extracranial veins. So they pierce the bone, the cranial bone. We have lots of emissary veins. I will show you some examples right now. But if you remember, we have discussed some of this when we learned the skull. Yes, for example, you can see here the condylar foramen and mastoid foramen. These are also openings for these emissary veins. Clinically, I have to mention the subdural hematoma. So this is one more type of traumatic brain injuries and one more type of intracranial bleedings. And it happens as a consequence of a rupture of a bridging vein, as you can see here again in this picture, after a trauma. And this trauma is not always a big trauma, in contrast to the meningeal, to the epidural bleedings. It can lead to a chronic subdural hematoma. It means that the symptoms will only appear after weeks of the trauma, and but this is also a very dangerous situation because, as you can see, it will also lead to the increase of intracranial pressure. And if you make a CT, you will see a crescent-shaped form, because here the bleeding is under the dura, so in this originally virtual space between the dura and the arachnoid layer. This table shows some examples for emissary veins. Yes, I underlined the most important connections. So naturally, you do not have to know all of these sites, only some examples. But clinically, they are also important because infections can spread, can extend into the intracranial space via these veins, because these are waveless and they do not have any elastic or fibrous or smooth muscles similarly to the dural sinuses, so they cannot collapse. So as a consequence, if you have an infection, the venous flow can return. So that's why if you have a wound here on the scalp, it can be dangerous. And at the end, some words about these diffusion barriers which are found in the CNS. We have mentioned the choroid plexus, for example, and this schematic drawing shows it. So this is also the inner blood CNS barrier formed by the endothelial cells, but as you can see, the endothelial cells are here fenestrated. We also have a basal membrane, and the barrier is actually formed by the ependymal cells, because here we have lots of tight junctions. We also have an outer blood CSF barrier. As I mentioned, this is the layer, the innermost layer of the dura mater, where we have these meningeal cells, which are also connected with each other by tight junctions, and they separate the extra meningeal vessels and the subarachnoidal space. And the third barrier is the blood-brain barrier, found everywhere in the brain, where we have the capillaries, yes? So the capillaries are also lined naturally by endothelial cells, as always, but here we have lots of tight junctions. So the endothelial cells are not fenestrated, but we have tight junctions between these cells, and we also have naturally the basal membrane, and this picture shows the doesn't show the astrocytes, but we have the processes of astrocytes as well around these capillaries, and it means that the substances cannot pass so easily through this barrier. And otherwise, here we do not have any other barrier between the brain tissue and the ventricles, and so the cerebrospinal fluid. It is only found in the choroid plexus. So these barriers have lots of biological functions. You can see some examples for these functions in this table. But in details you will learn about it in physiology. And we also have some places where we do not have any barrier, for example, in this endocrine glands of the brain, in the pituitary gland, more exactly in the median eminence and in the posterior lobe, where the hormones produced by the neurons must enter the bloodstream, or in case of the pineal gland as well. Or we have some other sites, for example, under the posterior commissure or in the area postrema. We also have, so we do not have any barriers. These are the so-called circumventricular organs. And in this case, we have another barrier, a barrier between the blood and the liquor. Okay, so I have finished. Thank you for your attention. And if you have some maybe questions, please ask.